Morning, ma'am. Silkworms teach us that beauty emerges from the simplest of beings, just as silk is spun from the humble cocoon. A very good morning to distinguished participants and our esteemed guests. Today, we continue our journey into the art and science of silk production, aiming to foster sustainable practices that enrich lives. So, with this inspiration in mind, let's embark on another day of learning and exploration. Ladies and gentlemen, it's a privilege to introduce our first esteemed speaker for today, Dr. Deepti Gupta. She is a remarkable presence in the Department of Textile and Fiber Engineering at the Indian Institute of Technology, Delhi, India, where she has held the position of professor since 2012. Dr. Gupta's academic journey is rich with accomplishments. She earned her PhD from the same department in 1995 and went on to join as an assistant professor in 1997. With over 35 years of teaching and research experience, her contributions have significantly shaped the textile and research landscape. Her expertise lies in the realm of surface functionalization, eco friendly finishing of textiles, microbe resistance textiles, functional clothing, and garment sizing. Her scholarly pursuits are well evidenced by her extensive portfolio of over 100 papers in reputable national and international journals. Additionally, she has edited 6 books and contributed numerous chapters to other publications. Guiding the next generation of scholars, Dr. Gupta has successfully supervised the completion of 10 PhD students. Her contributions extend beyond academia, as her work has been recognized by the prestigious Stanford University list of 2% most cited scientists in the field of materials for 3 consecutive years, 2020, 2021, and 2022. Innovation is ingrained in her journey. Her team's creation, the Mama Pod, an assistive garment, received the Biotech Product and Process Development and Commercialization Award 2020 from the Department of Biotechnology, Government of India. Beyond her academic commitments, Dr. Gupta's influence spans leadership roles such as Associate Dean of Infrastructure and Associate Dean of Students Affairs at IIT Delhi. She also serves as an independent director on the board of directors of companies, advising them on corporate governance. It's with great anticipation that we welcome Dr. Deepti Gupta for an online session to share her expertise on extraction and application of Sericin biomolecule. Her profound insights promise to further enlighten our understanding of this intricate field. Over to you, ma'am.
Thank you very much. Thank you very much for the kind introduction, and I'm sorry for the delay in joining. I'm very thankful to Dr. Tuteja for giving me this opportunity to address this gathering, and Dr. Seema as well. So, as has been said, I think you are all from the Department of Zoology, if I'm not wrong, and your interest is primarily in the perhaps in the sericulture aspect of it and the insect part of it. What we do is actually called as post cocoon processing. So we work in the segment. There's a lot of activity research that goes into rearing of silkworms, and finally from extracting silk from that silk yarn from silkworms. What we do, like I said, is a post cocoon processing. So once you have the cocoon, the process of making it into fabrics, which are used by all of us, is the area that I work in. And my focus of research for the past many years has been on, like was said, on eco-friendly finishing of silk. So just to give you a brief, because you are not from the textile background, textile manufacturing process comprises of several steps. Step number one is fiber manufacture, where you make the fiber. If the fiber is natural in origin, such as cotton, wool, silk, jute, linen, flax, we extract it from either the plant or the animal sources. And there is a series of steps involved in this process of extraction, right? And however, if I may mention, that 70% of the total trade that happens in the field of textiles is in synthetic fibers. Synthetic plus what we call as man-made fibers. So there is very little, and 30 is cotton, about 30% is in natural fibers, and the chunk is about 24-25% is in cotton in among natural fibers, and the rest make up 4 to 5% make up the entire other bouquet. So first step, as I was saying, is in textile manufacturing is fiber production. So it can happen from natural sources, it can happen from synthetic sources such as nylon, polyester, acrylic are synthesized from petroleum based sources. So that's step one. Step two is the conversion of the fiber that you get into what we call as yarn or dhaga, as it may be known as commonly. There is a lot of technology involved in converting these fibers or filaments into what we call as yarns. The primary process that happens in this case, some of you may be aware, is that we twist the fibers. We bring these yarns and fibers in the raw form together. We straighten them out, carding, combing are the processes, and then we twist them together. Without twist, it is not possible to weave the yarn. Right? Twist is what gives strength to the fibers and yarns. So there are many technologies that are used. So that is in our department also. The second group is yarn manufacturing group, which looks after the process of converting fiber into yarn. Then the yarns are converted into what we call as fabrics. So fabrics are of three types. Fabric is woven fabric. Most of us wear woven fabrics, but there are knitted fabrics, right? All your t-shirts and things like that are made up of knitted material. So knitting is another technology for fabric manufacture. Weaving is there. And third technology is non-wovens. So all the tissues that you use and all the disposables such as PPE clothing that was being made, the masks that were being made, they are all non-woven fabrics. So we have three main technologies in non-woven. After that comes the process of what we call as textile chemical processing. I am a part of that group where once you have that raw fabric, so the fabric that comes out of a mill is actually not possible to be used for apparel applications. We cannot use it for making clothing because it is not white, it is not absorbent, it's not finished, it is crude or raw. So all the sequence of processes that we apply after the fabric comes out of the loom is called as dyeing, printing, and finishing. So that's where I work in. That is looking at, so finishing is the last step in a fabric. Finishing doesn't mean finish, doesn't mean khatam kar do usko, but finish in textile terminology means value addition. जहाँ हम उसको वाइटन करते हैं, जहाँ हम उसको absorbent बनाते हैं. But more importantly, nowadays we have very important finishes such as, for example, flame retardant finish. So firefighters and all the carpets that you have in an airport or in a hotel, all the curtains in a hotel, all the car seat covers, all the seat covers in an aeroplane have to be flame retardant. I'm sure you have heard about the incidents of fire nowadays indoors, and that is making the legislation for making these textile materials flame retardant. So flame retardant is one type of finish. Antimicrobial is, for example, another type of finish where we try to, nowadays, recently people say, I don't want my clothes to smell bad, you know. So, or I don't want to catch infections from people. So antimicrobial finish has become a very, very popular finish now, particularly, for example, in socks. आपने देखा होगा कि your socks start smelling, particularly in this hot, humid weather. Either there is fungal or microbial growth, or they start smelling. In order to avoid that, why this odor happens, why this smell happens, is because although sweat is odor free, there is no smell in sweat, But when the microbes present on the skin interact with the sweat, so sweat has salts and sweat has dead skin, which is the nutrient for microbes. When these bacteria feed on the components of sweat, the product of their metabolism is the generation of odor, which we say as bad smell or body odor or bad smelling shoes or bad smelling socks. I'm sure all of us have experienced this traveling in metro or in crowded places. So people are now saying, give my textiles an antimicrobial finish. For example, in America, all the millions of pieces of socks that are bought for the military, American military, have to be mandatorily antimicrobial finished, right? So that even if they wore it for 1 week or 2 weeks, the socks will not smell and they will not develop any infections in their feet. So this is the area where we mean by finishing. I just wanted to give you, so there is flame retardant finish, there is antimicrobial finish, there is wrinkle free finish. And now we have a very new category of finishes, which was not there about 20 years ago, which is called as wellness finish. So, for example, you must have seen in the media, there is a lot of this thing, moisturize your skin, anti-aging, cosmetics, anti-aging moisturizers and things like that. So earlier we were putting it all on the skin, but now they are saying, why don't you put them on our textiles? So if I wear this should give me some nice smell, which makes me feel fresh, which makes me feel energetic. There are claims that if you put certain kind of amino acids in your clothing, then they are slimming. So you don't have to go to the gym, you don't have to work out, but ये कपड़ा पहन लो और automatically slimming होती रहेगी. So that are the kind of developments or expectations that consumers have in the field of textile finishing. And that's the area I work on. And I work only on natural products, which are waste or byproducts of some industry, and use them or develop the technology for applying them on textiles so that we can impart these properties. So today what I'm going to talk about is related to this. Please allow me to share my presentation. Just give me a minute, please. Can you see my screen now?
Yes, ma'am.
Thank you. Can you see it now as well?
Yes, ma'am.
All right.
Yes, ma'am.
And please
So,
make it on the slideshow.
yes. Okay. Is that okay?
Thank you, ma'am.
Yes,
Yeah.
ma'am. It's visible now.
All right. So one of the this work we did quite some time back, and that was the time when the awareness for eco-friendly textiles, actually textile industry in general, is under a lot of pressure because it is considered to be one of the most polluting industries in the world, right? So with given this increased interest in eco-friendliness, carbon footprint, sustainability, eco-friendly, what is happening to the environment, there is a lot of pressure on textile industry to actually clean up its act. And textile processing, of which I'm a part, is actually the maximum under the radar because all the chemicals that we use, all the water that we use, all the energy that is used is very, very polluting in its nature. So therefore, uh, there was so like we are on the topic of silk today. silk, uh, let me begin with this. When the raw silk is extruded by the silkworm, I'm sure you're familiar with this. This comes out in this form. So, what is the silk raw silk filament as it comes out is in this form. This is the scanning electron microscope. What you can see is that this is composed of uh, two triangular segments, which are the silk fiber or yarn, which is made up of one type of protein. I'm sure you know about this, is fibroin, right? So this is the silk yarn that we make saris and dresses out of. However, and it is extruded as two filaments together, right? These are the two filaments together. The raw silk fiber is called as beef, and it consists of two fibroin filaments. Each one of them is called as brin. Okay, and as you can see, the cross section of silk filament is triangular, and this is one of the major reasons for the luster of silk. This triangular cross section, it is what gives silk its unique luster. However, these two brins are covered with what you can see a resinous kind of material, which is also another type of protein, and it is called as sericin, right? And this is a gluey, gummy kind of a material which coats the silk yarns in a very thick film. The composition of sericin, maximum sericin that is present, of course, it varies in various varieties of silk, etc., is 25% sericin, and the rest is 75% of fibroin. Now, if we look at it chemically, right? So fibroins are also protein. Fibroin is protein, and sericin is also protein, but they are very different in their chemical structure, and they are very different in their properties. So while fibroin comprises a bundle of microfibrils composed of ordered amino acid sequences, this makes it a highly crystalline material which is water insoluble. On the other hand, sericin is the outer, as we saw, globular protein comprising of both random coil as well as beta sheet structure. I'm going to talk more about sericin here because that is the focus. And this is what makes it special. It is made up of 18 amino acids, and the maximum percentage you see is 32% of serine, and it is water soluble in nature. The other important properties, as far as we are concerned from the textile finishing point of view, is that it contains a lot of polar groups, including hydroxyl, carboxyl, and amino. Why this is important is, unlike fibroin, for example, which is crystalline and which does not have many free polar groups, this allows us to play with the macromolecule. It allows us to graft it, it allows us to apply it to different materials. And this bonding between different materials is only possible if we have this kind of uh, groups. The hydroxyamino acid content in sericin is about 46%, along with 42% of polar amino and non-polar. Now, actually, for till almost let me say about 30 years ago, silk has been fiber that has been known and processed for hundreds of years, this thing. But uh, sericin was the discarded part, and it was only 30 years ago that people realized that sericin also has a lot of important properties. So people were only focusing on fibroin, extracting fibroin, and discarding the sericin. Nobody really studied sericin till about 30 years ago, and for the first time in Japan, when they started looking at sericin also, they realized that it is a wonder molecule, it is a super molecule, and it has a lot of therapeutic properties. So it was only about 30, 35 years ago that people started researching on sericin as a biomolecule, and now of course it's a very expensive, more expensive than fibroin as a matter of fact, and being used in food sector, used in um, pharma and all such applications. So this is another, just to show you that they are not very different in their molecular weight. This is sericin and this is fibroin, and this has a lot of polar groups like I mentioned earlier. So what is it like when the silk is processed? Actually, the first step that is done is called as degumming of silk. So like we said, sericin is a gum, fibroin is the filament, and sericin is the gum which glues these two brains together and encapsulates them. So when you take out sericin silk from the silkworm and reel it, you get something like this, which is the raw silk yarn. As you can see, it is yellow in color. What you cannot see or feel is that it's highly stiff, it's highly rough in nature, and it is not lustrous. So the first process that is undertaken in the processing of silk by the silk processing sector is what is called as degumming, or in other words, removal of sericin or separation of sericin from fiber. Once the silk yarn has been completely degummed, you can see what happens to it. It becomes white, it becomes soft, and becomes lustrous. And only then can it be used in manufacturing of clothing. You cannot use this yarn because it's too stiff, and every time you will wash it, some of the sericin will keep coming out and the properties of fabric will keep changing. What silk is known for is this kind of soft, lustrous look. So the first process, like I said, is removal. Once you remove sericin, so this is the process that has been studied extensively for many, many years, right? So the process is of cleavage of the peptide bonds between fibroin and sericin. So there are many methods. I'm going to focus and talk about methods subsequently. Basically, what we are trying to do is, here is the raw silk as it is extruded with the two fibroin filaments encapsulated in sericin, and we subject it to a process called as degumming, which basically breaks the peptide bonds between fibroin and sericin and gives us these clean, clear filaments, right? So the cross section can vary between cultivated silk and white silk, but the process essentially remains the same, right? So first we use some method. I'm going to talk about this method. The methods are basically hydrolytic in nature, and they break the bond between fibroin and sericin. And once this bond has been broken, material is subjected to extensive washing to remove this solubilized sericin, right? And pure fibroin is left. And pure fibroin, as we know, has this lustrous and soft look. So there are many methods which have been used traditionally for degumming of silk over the years. And the one that was most commonly used in Europe, because Europe was, in fact, let me just mention, is that Italy in the world has been the leader for production of very high quality silk fabrics and dresses from silk. So you may know that Milan has been a fashion capital of the world for almost a century, and the best silk processing happened there. What they used, the traditional method that was always used for degumming of silk, because silk has never been a mainstream fiber, silk has been a luxury fiber, right? So only very small amounts have been processed, and you create very, very highly fashionable and very highly exclusive kind of garments. So they were at that point of using soap, a kind of soap which is called as Marseillese soap. So they had optimized and find one type of soap which is an oil based. It is actually an olive oil based soap. So you can imagine how expensive it would be. It's a very expensive soap. that the industry used for a very, very long time. And you see, you are using almost 20 to 30% soap on weight of fabric, right? A very large amount. Subjecting it to boil for 2 hours, and you would get an yield of about sericin removal of about 19%. So for years and years, this was used, but when other countries started producing, they realized that soap was a very, very expensive way of. It gave the best quality silk, but it was very expensive for large scale production. So people tried water. Of course, water is the most efficient, and the process is called as HTHP, which is high temperature, high pressure processing of silk. And silk would be treated at, now see, the temperature has gone up from 100 to 115, and the time has gone from 2 hours to 3 hours. So you need a very, very intense processing of silk material at high temperature for a very long time to get what you want, right? But as you can see, that using this temperature for this duration of time is very, very energy intensive. So therefore, this is also an expensive method, and the quality of silk would not be so nice because of if the water was hard and so on. So not very effective. People would prefer to use this, but not effective from point of view of quality of silk obtained. People have used alkali also at a pH of about 10. Now see, the temperature has gone down. When we add chemicals, the temperature has gone down, and um, the time has gone down significantly, right? So from 3 hours, we are at 20 minutes, and you get removal of nearly 24% sericin, right? People have also used enzyme, because in textile we have always had this using of biotechnology as a substitute for chemical processing, and a lot of proteases and other enzymes combinations have been used, and you get this kind of yield. I'll talk more about this comparison in a bit. More recently means also about 20 years ago, organic acids people tried, right? And you see the pH is low, temperature has gone down, time has gone down, and then amines have also been used. So these are the conditions which people have optimized for various processes. If we look at the comparative study of these degumming methods, so in soap, we have all these options available. How soap works is that it forms a chemical bond with sericin, produces soda salt, basically an emulsification process, right? The advantage is that the luster is good, and the strength is good, and the only problem that happens is that soap, if the water is used is hard, so you need soft water for this. It leads to formation of insoluble metal soaps, which may redeposit on fibers and interfere with the appearance and luster of the material. In alkalis, all these alkalis and their combinations have been tried. Process is that of hydrolysis. Productivity is high, processing cost is low. This is the cheapest and easy to handle, but the fabric that you get is yellowish in color, and therefore you have to use some buffers, etc. Acids, all these acids have been used. The acid is specific in it. The good thing is that the acid is very specific in its activity and attacks only the aspartic and glutamic linking peptide bonds. So therefore the degumming is very quick, and we save on thermal energy and time. However, acid may damage the fibroid. Then enzymes, trypsin, papain, variety of proteases have been used. Again, the nature of enzyme is very specific. It attacks on the peptide bonds present between carboxylic acid of arginine, lysine, histidine, and so on. The fibers are less prone to structural and surface damage because there will be no effect on fibroin. The only action is on sericin. the temperature and pH conditions are mild. However, because of these mild conditions, complete removal of impurities doesn't happen. So the feel that you get after enzymatic degumming may not be quite as desirable. The last and the most recent was, amines, use of amines. And this is also specific in nature. It cleaves the peptide linkages between sericin and fibroin and gives uniform undamaged cell fibers. It's not affected by hard warp. But the rate of degumming is low. So this is the kind of, comparison. There are many methods, and so every processor, every industry has to choose, you know, whether they want to have an energy intensive or if they have soft water, how much do they want to invest in the cost of energy, water, and chemicals or enzymes used. All these methods are available. So a lot of research, this was just to tell you that people continue to work on these methods just to get the right quality of, right yield and quality of silk from the um, extracted this thing. So I've already talked about this. These are some of the comparisons of this. Now, more recently, in the last 20 years, people, because we've seen ki out of all the methods that we talked about, none is like perfect. किसी में कुछ problem है, किसी में कुछ problem है. So people continue to work on these because now new technologies, I'm sure you are aware of these, such as supercritical carbon dioxide, microwave, and ultrasonication have now become available. Now, these are various forms of energy, right? And they are more effective, than thermal energy. So, in almost all our textile processes, we are making use of these technologies, and we call them as physical methods as against chemical methods, right? So, you know that in order to catalyze any reaction, you need energy, right? So rather than thermal energy, can we use these relatively newer and more effective and cleaner? These are also called as cleaner technologies for all processes. So they have been used for degumming of silk as well. Very interesting. So water continues to be of interest. We always try to find if we can do something with water. It's the most eco-friendly and the cheapest resource. So people have electrolyzed water, and it is called as strongly alkaline electrolyzed water, which has been used for degumming of silk. People have also used urea with buffers. People have used supercritical carbon dioxide. It is being used for dyeing also now. People are trying to, because it's completely dry. No water is used and no effluent is there. So supercritical carbon dioxide technology, I'm sure you have heard about it. Then microwaves, all of you have heard of it. We use it in our kitchens, but it's a very useful means of energy now in the radio frequency range. In textile also, we use it for various purposes to, activate reactions. Ultrasonication, of course. So in your labs, I'm sure you're using these things. A little bit about these processes. So strongly alkaline electrolyzed water, in one study, they synthesized it by electrolyzing the tap water. And what they did by this process was that the hardness of water was reduced by 30%. So you see, we talked about that the hardness of water creates a problem, right? And then um, using this electrolyzed water, they were able to completely remove sericin in 20 minutes. Look at the reduction in time. pH is still a bit high, but that's all right because the time is very, very low. And they were using this electrolyzed water in a ratio of 40 to 80, mixing it with normal water. And degumming rate was higher, as we can see, very quick. And mechanical and thermal properties were same as those which we get with the best option, which is soap, neutral soap, right? People have also tried urea, 8 molar urea with buffered with tris SO4 and mercaptoethanol, etc. 2 hours, 80 degrees. Temperature is much lower. And mechanical properties, as you can see, are even better than SAEW and much better than that of alkali. So this is how every method is compared with alkali or soap to benchmark it. Ultrasonication. Now, ultrasonication and microwave, by the way, are not used independently. They are not being used in a water medium. So they are being used in combination with one of the traditional methods, such as soap, alkali, acid, or enzyme. So you treat your material with soap, alkali, acid, or enzyme as per the previous traditional technique, and then subject it to either microwave or ultrasonication, or for that matter, SCO2 also, right? So that energy you are saving on, temperature you are saving on, water you are saving on, right? But we are still using the same chemistry that we understood earlier. So ultrasonication, as you are aware, is based on the acoustic cavitation effect, where highly energetic, high energy molecules are used to break larger molecules or larger aggregates into smaller parts, and therefore it directly acts on sericin, and that's how degumming happens. It cracks and separates sericin without any damage to fibroin. So obviously the processing time is reduced and the removal of sericin is more uniform. Similar is the case with microwave assisted. We supply high intensity energy and degumming by SCO2, etc. Also. So this is a flowchart that shows the process. So you take the cocoon, you discard the larva, cut the cocoons, treat it with any of the traditional agents such as alkali, acid, or enzyme, and then subject it to either ultrasonication or microwaving to get degummed silk. So I would like to stop here now, maybe pause and take questions, if any, before I move on to the work that we have done on degumming of silk and application of silk-based finishing. If there are any questions, I'll be happy to take them.
The house is now open for questions. If anybody has any questions, please, you may please ask, ma'am. Ma'am, I believe there aren't any questions. It is really clear, very
Okay.
simple to
All
understand.
right. Yeah, because this was a separate section. Now I'll focus on our work. What we did, this was the kind of background about what degumming of silk is. So, okay, so I'll continue. So, what basically, when we started working on this, what were the problems? You see, all this that I have talked to you about was focused on removal of sericin, right? Removal of sericin and fibroin. However, all the focus was on fibroin. What kind of silk yarn were we getting out of it? And sericin was being simply discarded. Now, when we started working, we realized that silk has, of course, it was being talked about that, or there was evidence, uh, scientific evidence to show that sericin is. So, what was actually happening was that when so much of processing was silk was happening in Japan, the sericin that was going out into the effluent waters was actually increasing the TDS and adding significantly to their pollution load. So they had to remove sericin from the water before they could release the waters into the main water bodies, right? And that's when Japanese scientists said, let us look at what we are discarding, let us see what kind of properties this molecule has. When they started studying it, they realized that it has a lot of medicinal and other properties. And therefore, that's when they started to recover sericin from the wastewaters and use it separately as another valuable material. सो अभी तक जो मेथड्स का मैंने डिस्क्रिप्शन दिया है उनका फोकस सिर्फ था टू गेट द राइट यील्ड एंड राइट क्वालिटी ऑफ सिल्क yarn. And those methods did not, I mean, nobody studied the sericin that was going out or what was happening to the sericin, because the only purpose was to remove the sericin. When we started working, our focus was to work on sericin. So we needed to have methods which would allow not just silk yarn to be produced, but sericin to be recovered and not damage the properties of sericin also. So in many ways, it was very different in the sense we didn't want to damage the silk, of course, that is the main product. But we also wanted to conserve sericin in its pristine form, and also to maximize the yield, right? So that is why we started looking at recovery of sericin. It is still degumming, but here the focus was on also maintaining the quality and quantity of sericin that we were getting. So we launched on a series of studies which were aimed at extraction in an eco-friendly manner, extraction such that sericin was not damaged, characterizing the sericin that we were getting, because not much was known about the properties of sericin at that point of time, and application of the extracted purified sericin on textiles to get some additional properties. So the question is, why sericin? Right? So like I said, it has some amazing features, such as it has antioxidant properties. I'm sure all of you have heard nowadays you should eat antioxidants, eat these fruits because they are rich in antioxidants, eat this because it is rich in antioxidants, etc. So antioxidant means actually something which preserves or enhances or preserves the, or let us say, controls the cell degradation, which may be called as an anti-aging process, right? So it maintains the cell health and controls aging, right? So antioxidation, bacteria, we already said, antimicrobial properties. We are also looking at ultraviolet light because we know it's highly damaging to human cells, and improved hydrophilicity. So wherever in moisturizing creams, etc., you are looking at hydrophilic agents, and also the material is biodegradable as against all the synthetic chemicals that are used for these purposes. This material is coming from the nature, is a waste byproduct, and is also biodegradable. So these are some of the salient features of the sericin biomolecule. It has also been shown in clinical studies that it has biological activities such as tyronase activity inhibition and pharmacological functions such as anticoagulation and anti-cancer properties. So it is actually being used as a drug carrier and a drug. It is actually being, sericin tea is now available for diabetics. It's supposed to be very good for diabetics. Sericin, if ingested, is also considered to be aiding in weight loss. And besides this, there are specific anti-cancer and anticoagulation properties. As we said, because of the presence of a lot of uh, polar groups, particularly the serine amino acid, it has very high hydrophilicity. So actually, human skin also is very rich in serine amino acid. And that is why the cosmetic products made from sericin are considered to be very good, because they maintain the or have the same function as human skin, right? So therefore, moisturizers and cosmetics, sericin-based cosmetics, are very, very expensive, being made in Japan, etc., and very popular. The actual biomedical application of sericin not been fully explored. because of the difficulty in processing it into useful material. So production of Sericin is not easy. We process tons of silk in India every year, and we are practically not producing any Sericin. We try to actually purchase Sericin for our studies, and nobody is producing that. It's very surprising. But on Alibaba.com, or Chinese companies are producing Sericin in bulk, and they are selling them all over the world for various applications, like I said, pharma, food, cosmetics, etc. But in India, there is not even a single producer, because even now production of Sericin in a large scale is really not, I don't know, it's technologically a bit challenging. So what we did was a series of studies we carried out. The first was extraction and characterization of Sericin from different sources. So there are many, many sources of silk, as you would know. There are mulberry cocoons, where the cocoon is there. Then there is something called as Sericin silk waste, right? So when reeling waste. So when you reel or make the yarn from the cocoon, there is some waste that is left. It is called reeling silk waste. There are silk flags, which are the cutout left cocoons, and there is the woven silk fabric, of course. So we wanted to see that if we want to get the best quality Sericin, which is the best starting source for us, right? Out of all these, we have the choice. We can directly extract from cocoons. We can use waste. It's a cheaper material. Silk flags are also cheaper, and woven silk fabric is the last stage. So should we extract at that stage to get the properties that we want? So we tried all, and then the Sericin that was extracted out was characterized. What we did was that we developed or applied Sericin on polyester fabric. Now Sericin, as you know, is a macromolecule, and polyester also, as you know, is an aromatic has an aromatic chain, and it is highly nonpolar, highly crystalline. It's not easy to apply anything on polyester in a durable manner. Your garments are washed every day, so we want the finish to be durable. So what we wanted to do was we wanted to develop what is called as cosmetotextile. Cosmetotextile मतलब polyester fabric is hydrophobic. आपने देखा होगा कि in this kind of hot humid weather in Delhi we can't wear polyester, right? Because it is completely hydrophobic. Sericin is hydrophilic. So can we apply a hydrophilic material on a hydrophobic material so that it becomes hydrophilic on the surface and gives you the comfort? We have also said that Sericin has antioxidant properties, anti-stat properties, right? आपने देखा होगा static generate हो जाता है जब आप polyester या synthetic material पहनते हैं, it goes chick chick chick, right? Because of lack of, again, because it is hydrophobic. So putting a hydrophilic material on that can actually improve the properties. And we also said that Sericin has antioxidant properties or anti-aging properties. So can we put a material on polyester when you're wearing is actually good for your skin? So normally they say polyester is not good for the skin, but polyester is a cheap fabric and it is light fabric. It is easy to wash and maintain. So can we add this biomaterial on it to impart the properties which are otherwise not there? That's what textile finishing is about, right? That you take a base polymer which has certain good things but lacks in other properties. We take these materials and put them on this to impart some value added properties. So this is a technological challenge, actually, how to add or apply a finish durably on polyester, right? So directly there is no affinity or no bonding can happen between Sericin and polyester fabric. So there are two approaches that are possible that we use in textile processing. One is to modify the polyester fabric in some way. There are various ways which we used in this study to create some functional groups, some reactive groups on the surface, only on the surface at the nano surface level, right? And then sericin also has polar groups. When I apply sericin, the sericin molecule binds to these uh, polar groups that have been created, and that's how sericin gets deposited. The other approach is to modify sericin, add some kind of grafting groups, etc., on sericin, which is possible because of the process, presence of polar groups, and such that they can bind covalently with the polyester fabric. What we did, because we wanted to use milder chemistry and we wanted to use green chemistry, we pre-treated polyester, right, to create these functional groups, and then applied sericin and then tested the fabric. So, pre-treatment of polyester, we tried with 3 methods. We wanted to compare and see which is the best method. The traditional method जो हमेशा use करते हैं इसको causticization बोलते हैं, weight reduction of polyester के लिए. We use it with alkali, right? So this is a chemical based, cheapest but chemical based method. The effect in all the cases is the same, right? It's only the let's say the eco-friendly footprint that varies. The second was enzyme, right? An expensive, eco-friendly, but an expensive option. And the third is physical irradiation. Completely green, completely dry, no chemical required, no water required. That is exposure to radiation. High intensity, in this case we used monochromatic 172 nanometer UV excimer lamp. तो बस आपने लैम्प के नीचे फैब्रिक को रखना है, and because of this high energy radiation, these same functional groups are created due to the cleavage of the bonds in polyester. We compared all these 3 methods to generate functional groups, and effect of each one of these treatment on the properties of polyester was studied and compared. The best method that was found was then used, and then we applied sericin onto this functionalized polyester. So surface functionalization is one technique which is now coming up big time, which is a green technique, which is a dry technique, and therefore we wanted to use that also. Relatively new development that has happened in the last 2 decades. And then we tested the developed fabric for various properties which we wanted to have, that is hydrophilicity, that is dyeability, what is the effect on dye uptake, anti-stat. So we said this is the problem with polyester, right? Static generation, and also antioxidant property, all these properties. So this was one study that we carried out. I'm going to talk about the results in a bit for what we did. We also, because of this medicinal properties of sericin, we also applied sericin onto cotton wound dressings, right? So we have wound dressings. You put various kind of things on wound dressings. Sometimes, your wound has an oozate, and you want to use a dressing which will absorb all the exudates that are coming from the wound. So if you have a highly hydrophilic material, and also a material which has antibacterial properties and such like that, it can reduce the infection, and it can, be absorbent also. So we developed that also. The other thing that we did was, because sericin is a high molecular weight polymer molecule, application on textiles is sometimes challenging, right? Because a large molecule cannot penetrate into the textile structure. So what we did was, we also did a study where we produced sericin peptides, right? So through enzyme hydrolysis, combination of enzyme and organic acid, and both we tried. We did not get very good results with enzyme, but we got good results with acid. And then we applied these to see whether, penetration and durability of the finish was better. Another study that we carried out was on shrink proofing of wool. And so I'm sure, I don't know if you have used, I mean, pure wool has become so rare nowadays, but if you have pure wool sweaters or shawls, you must have seen that you cannot put them into a washing machine because they shrink. So this is a characteristic property of the wool fiber because of the presence of scales, that they have a differential frictional effect, and the moment you wash them, it shrinks. I'm not going to go into the detail, but I'm sure all of you have experienced it. So there is a demand in the market for making wool shrink proof. Can we make wool shrink proof? The process that is used for shrink proofing of wool is two-step. It is one case we modify the scales, they're very sharp scales. And the sharper is the scale, the more is the propensity to shrink. So if we modify the scales or damage the scales in, partially, then and coat these modified scales with a polymeric coating to make it smooth, it makes the wool shrink proof. So shrink proof wool is used, but the process that is currently used, or the traditional method, makes use of very strong chemicals. It's a chlorine based process and makes use of synthetic polymers. What we wanted to see was, can we use uh, eczema as UV radiation as a substitute for chlorine based scale modification, and instead of the synthetic polyurethane polymer, which is non biodegradable, can we use Sericin as the polymer to coat these modified scales? So we developed a more eco-friendly substitute. Both processes were substituted with more eco-friendly, less use of chemicals, less water, less energy, and biopolymer to make shrink-resistant wool. And then a study that we also planned, and the chemical engineering department actually did a very successful study on this, was to use Sericin, make filters which were coated with Sericin, and this was used for absorption of water cleaning, with water cleaning properties. So it can also be used for removal of drugs and dyes from water. So these were some of the studies. I'm just going to talk about one or two of them in detail, the first ones. The rest, these are uh, publications are there. I will share the list of publications with you. For details, you can refer to those. So what we did was that we first thing that we did was to produce Sericin in large scale, and like I said, the interest was in using the water-based method. The moment you use alkali or soap or amine or anything else, you have to again separate Sericin from that. तो सेरेसिन तो आपने रिमूव कर लिया, पर जो आपके पास वेस्ट वाटर आया उसमें सेरेसिन भी है और उसमें सोप भी है और उसमें अल्कली भी है. That itself is a very complex process of removal. So we wanted, because we wanted to recover Sericin, we wanted to use a method which was pure water based. So what we did was we tried four, actually not two, in this it is shown as two, but we used four sources of silk to extract Sericin in a water based medium. We used HTHP, which is high temperature, high pressure, about 120 degrees, 130 degrees of processing, and we used infrared heating. So this was our main novelty that we tried to use, a new form of energy, where IR, which is a more uniform and a quicker means of heating a system, which was also available in our lab. So we compared both these methods for, seeing the effect on the quality and yield of this thing. We got a liquor. This liquor was actually then subjected to two kind of drying methods. One was spray drying and one was tray drying method. So we were trying to develop a method for large scale production of sericin, whichever was the most effective. So we took 4 sources, 2 methods of heating and 2 methods of drying to get the sericin powder, which was then subjected to characterization. We did SEM, we did SDS page, we calculate the yield, the protein content, we did X-ray diffraction to see the crystallinity. We looked at the ultraviolet spectra because in proteins that's the standard method of looking at the quality of material, subjected it to fluorescence spectroscopy, FTIR spectroscopy, etc. to characterize the material. So this is what our process looked like in a graphic form. Extraction, then we kind of subjected it to tray or spray drying to get a powder, and the powder then was subjected. What we found was that the method of drying actually can also, you see the morphology of sericin that is obtained can vary with the method. If you do use freeze drying, the crystals look different. If you use tray drying, they are different. If you use spray drying, they are different, and the properties are also different. So these were the 4 sources that we used: cocoons, silk waste, flax, and fabric, right? And we extracted silk by 2 methods in this. And as you can see, a simple extraction, and we used UV spectra extensively just to characterize our silk. And what we did was we bought a pure standard to be used as a from Sigma Aldrich, which is called as standard sericin. So we used this standard sericin to benchmark our the samples that we were extracting. So considering that to be the gold standard, what you can see here is that the blue, which is the gold standard, the graphs obtained from each one of these 4 sources are different. So one thing was established, that you get different types or different quality of protein if you use a different source. What you can see here is the fabric and the cocoons are yielding the best quality sericin. So we continued further with fabric. Anyway, fabric is something which is accessible to us, and in our department we work with fabric. And the quality of silk that was coming out of it was also the best. So therefore, sorry, the rest of the study we continued with this. So this is the kind of results we got. So as you can see, what we did was we used 2 medium of heating, that is IR and HTHP, and we used 3 temperatures with both these methods. So 100, 110 and then 120. And we studied the effect of time of extraction on the yield of sericin. So yield of sericin is very important, right? The maximum yield that we can get, and we said it's about 25% is the target, the better it is, right, for us, because we need to extract sericin. What you can see here is that at each temperature, one finding that is there is that in at all the 3 temperatures, 100, 110 and 120, these are the 3 graphs. The lower graphs are for HTHP method. So you can see very clearly that the amount of sericin extracted at all the time periods is lower with HTHP. The yield is going up as the temperature is going up. You can see that maximum yield is at 120 degrees. So if we want to use HTHP method for extraction, we have to use 120 degrees, 120 degrees, that's right. And we also need to treat for 120 minutes. That's when we get the maximum amount, or 90 minutes also maybe, we get the maximum yield. However, what you can also see is that in all the temperatures, at all the temperatures, the yield is higher for the IR method. It's very clear. And look at the difference from, if I look at 60 minutes, for example, Here it's about 8% yield, and here it is 26% yield. So you can see that with IR, I can actually extract much more sericin at 60 minutes. And I really don't have, there's no change after 60 minutes. So at a much lesser time, which is energy saving, I am able to get much more this thing as compared to the conventional HDHP method. So that was our first major finding, that the kind of system that you use, and reducing the temperature from 120 time from 120 to 60 minutes is a significant saving in energy. So this paper actually got accepted and published in the Journal of Cleaner Production. So we were able to show that the method, IR based method, is much more clean as compared to HDHP method. We also studied the effect of drying method, like I said. Now you can see very clearly the amount of protein present in standard sericin. आप देखिए 1.73 था. IR, now we are only looking at IR 120 60 minutes sample. Look at IR spray and IR tray and HDHP spray and HDHP tray. So much protein loss is happening just when you change the method of extraction and method of drying. Right? So obviously it's clear that IR spray is the best method. So we were able to establish that. And here again, then we were able to look at the UV spectra of standard sericin, right? And then you can see that with tray and with each method, uh, the spectra is different, which means some property or the quality of protein is being affected. We have ensured that. Let's look at overall comparison of these two methods. Yield was 23% in at 100 degrees 40 minutes for IR, and 8.4, significantly lower, right? And the protein quality assessment, as you can see, standard sericin is this much, because standard sericin we believe is freeze dried and things like that, so better retention, but this is not so bad. And then protein, if you see content, less than standard, but much higher than HDHP. Ash content and moisture are this thing. So this is how we characterized and benchmarked the two types of sericin extracted by different methods with standard sericin. Here also, like we see, A is standard, which is also freeze dried, most likely. This is spray dried. You see, it's very different spherical balls. And we also just attempted to, carry out freeze drying, and we find we get the kind of crystals that are there in the standard sericin. So this is the sericin that we used for this thing. Similarly, of course, effect of, so then we further studied, the effect of, can we lower the temperature further? We working at 100 and so. So we wanted to see in IR, can we go lower? But the extraction, as you can see, is not significant. So we stuck to 100 degrees. Oh, sorry. So this is the next study. So once we had got our sericin extracted and characterized, we now wanted to start the process of applying it on sericin, on polyester. So this is the kind of flow chart that we used for this process. So we took polyester fabric, surface modified it. Like I said, we used three methods: chemical, NOH, physical, using 172 nanometer UV excimer lamp, and biochemical or enzymes such as lipase and glutenases. Then this modified polyester, on this modified polyester, we applied sericin. So extraction of sericin is there. We applied Sericin and then characterized the Sericin treated polyester fabric. So what we studied was weight add-on, dye assay, fiber morphology, bending length, moisture absorption, etc. And in terms of properties, we wanted to study the anti-stat property, antimicrobial, ultraviolet protection, and radical scavenging activity. Right? So out of the three methods that we used for modification, we found that UV excimer lamp was the most promising. Why? Because excimer lamp is just a lamp, switch on, switch off. So you don't need a bath, you don't need a padding mangle, you don't need water, you don't need anything else. It has a very large emission area. Cost of the lamp is high, but then subsequently cost of exposure is low. It's a dry process, simple handling, no effect on bulk properties. The only modification that is happening is at nanometer, 9 to 10 nanometer, level of the top of the fabric gets modified. So the rest of the fabric remains unaffected, unlike chemical, which enters the bulk of the polyester and lowers the strength of the polyester, etc. There's no thermal damage, temperature is not raised. So what was then was Sericin was applied on excimer modified PET by pant dry cure method. Add-on, that is 1.5% of maximum Sericin we were able to apply. So like I said, applying one polymer on another is not easy. So textile is a polymeric substrate, and then you want to add something to it, it's a big challenge. And if you add more than 1.5 or 2% of anything, the fabric becomes stiff, the fabric becomes heavy, the permeability of the fabric goes down. So we really don't want to apply too much. So 1.5 is a very optimum kind of this thing. We had to use 10 grams per liter of Sericin at pH 5 to get this add-on on modified polyester. Unmodified polyester पे तो हम इतना भी नहीं लगा पाएंगे. And finally, we cross-linked this thing, because in order to make it durable, you have to bind it covalently. So we used 10 ml per liter of glutaraldehyde and cured it for covalent bonding. Right? So this is the schematic. This is the kind of lamp we have, right? And the lamp kind of emits photons, to which polyester was this thing. There is oxygen-rich groups created. There is an oxygen cycle, ozone oxygen cycle that gets created in this space, and which leads to formation of oxygen-rich groups on polyester, such as these that you see. And then on this, Sericin is applied, a cross-linker is applied, and you are able to durably create a polyester fiber, which now has a coating of Sericin. I think I'm going to skip this. I've already kind of talked about it. We characterized our surface, to see what was happening to the surface of the polyester fabric. So this was the control fabric without any this thing. Alkali treated fabric, देखिए कितना damage हो गया, you can clearly see. So alkali creates, of course this is at nanometer level, but there is a roughening of surface grade. So this roughening actually allows deposition of coatings, facilitates the deposition of coatings on this material. And then if you see the excimer treated, देखिए कितना uniformly और कितना small narrow, इसमें देखिए कितने बड़े बड़े damage create हो गए. So the treatment can never be so uniform. Look at this, what happens with excimer. It's absolutely, the surface has become rough, right? As compared to this, which is untreated. If you see, there is a nanometer level roughness. So excimer or radiation, actually what we say, creates a physico-chemical effect. I showed you the chemical effect in the previous slide. This is the chemical effect, right? That is, functional groups are being created. But AFM shows us that there is a physical modification of surface morphology also that is happening. So together with this surface roughening and the chemical functionalization, this is what allows a sericin to get deposited uh, on this thing. So you will not be able to, if I try to apply sericin on this, I cannot apply it. It'll just get washed off. The moment I wash off the fabric, it's going to wash off. Therefore, I need to create this physico-chemical functionalization to do that. So this is again the same thing that we see here. We did the same thing with this thing. I think I'm running out of time. I'll just quickly go through this. So then what did we test? Sericin treated polyester. What we found was that there was 4 times increase in moisture regain. Remember, we set out to make the polyester hydrophilic, right? So we get 4 times moisture regain. There was 70% increase in vertical wicking and moisture absorption capacity, right? And with time we measured the wicking. आप देख सकते हैं कि eczema treated and untreated fabric, right? So this is the kind of enhancement or improvement that we could bring about in the properties of polyester that we set out to do. Then we also said we wanted to make it anti-stat. So anti-stat के लिए कि cigarette ash test होता है, right? So you can see that on untreated polyester fabric, इसको लाते हैं and मतलब untreated polyester fabric is brought close to ash, cigarette ash, and you see a lot of ash sticks to it because of its anti-stat property. But when we treat the fabric, you can see that very little ash is. So this is a standard test for anti-stat property of fabrics that we carried out. There was 2 times increase in ultraviolet protection factor. So the material has become UV protective. You can go out in the sun, you can make tents out of it, you can make umbrellas out of it. It has acquired protective properties, and 40% improvement in radical scavenging activity. Radical scavenging activity is a measure of the antioxidant property of the fabric, right? So we found that the material is actually showing active antioxidant properties also. And all these properties together make it suitable for sportswear and such, because it is now hydrophilic, it's more comfortable, it's also used for medical, applications and so on and so forth. So basically, like we set out by saying that polyester is non-polar and hydrophobic in nature, to overcome these sericin application was required, and we cannot, because polyester is inert, we cannot apply any finish directly on it. So surface modification was carried out to incorporate functional groups which increase and improve the adherence of this thing. Then I talked about, I'm just going to quickly go over these. Preparation of sericin peptides. We also tried doing this using tartaric acid to create lower molecular weight, peptides. And we could also, it's easier to apply them on textile. And the last thing that we did was sericin based process for shrink proofing of wool. So we modified the scales. Wool fiber looks like this under the microscope, very sharp scales. When we treat it with UV excimer, you can see the scales have become blunted, right? The sharp edges are gone, right? And then we coated it with sericin. To 2-step process, 1 dry process, treatment with UV excimer, which modifies the scale, and then subsequent treatment with sericin, and the material became, shrink proof. We did this also, application of sericin on this thing, and then I think that's all. Thank you very much. And these are the, in case you are interested, these are the publications that we made out of this, and all the details of the papers are available. Thank you very much for a patient hearing.
Thank you, ma'am. Your insights have truly deepened our understanding of silk fiber extraction and usage of sericin in textile processing industry. Ma'am, we have some questions from the participants. Actually, there was some problem with the mic. You asked previously, but they couldn't unmute their mic. So
Yeah.
please raise your hands if you have any questions. Your mic is unmuted. You may go ahead with your questions. So you aren't visible. Get up.
Hello, ma'am. Can you hear me?
No, can't hear you.
Sir, could you please come closer to the mic?
Yeah.
Hello, ma'am. Can you hear me?
Yeah. Now.
Yeah. Just want myself, Dr. Rahul Dev. I just want to ask the anti-cancerous property of the sericin. Has it been worked out? Some experimental data is there? What kind of cancers on which they are being tested?
Yeah. So actually, lots. That was not our focus of study. But if you search for anti-cancer properties of an anti-tyrosinase property of sericin, there are a lot of clinical studies available. So it has been demonstrated very well. Of course, for textiles, we were not interested in that activity, and but it's been there, and sericin is being very actively used in anti-cancer treatments, etc. So
Thank
you
you,
can
ma'am.
find a lot of references. Yes, it's been clinically proven.
Do we have any more questions from anybody? If you could please either raise your hand or get up because you're not visible in the camera. Do we have any more questions? I think that's from the participants. We extend our heartfelt gratitude to ma'am for an enlightening and captivating presentation. Your expertise and passion for this field has left an indelible mark on all of us. Let's give a big round of applause to ma'am for today's presentation. Thank you so much.
I would have been happier if there were some more questions. No questions means everything is clear or not, no clear.
Everything
Everybody
is
was,
clear. It was a very, very nice presentation.
I thought it was something different from what you normally do. So there would be more curiosity and more questions, maybe. But it's all right.
Thank you, ma'am. Thank
Yeah.
you so much.
Thank you very much. Bye bye.
All right, before we move ahead, let's take a short tea break. I request you all to join for the tea at the canteen. And after that, we will resume in 15 minutes here itself in the committee room itself.